All right, let's jump right in. We're going to talk about something that completely changed the course of human history: penicillin. You've probably heard of it, but the story behind this miracle drug is just wild. It's a story about a lucky mistake, some brilliant minds, and a whole lot of industrial power, all kicking off with, well, a bit of fuzzy mold.
It's actually kind of tough to wrap your head around this, but think about it. Before the 1940s, the world was just a lot more dangerous. A simple scratch from a rose thorn, a scraped knee, that could literally be a death sentence. There were no antibiotics, so common bacterial infections could, and often did, kill you. It's a wild thought, right?
And that's what makes this first part of the story so amazing. It's truly an accidental miracle. See, the discovery of penicillin wasn't the result of some grand plan. Nope, it was a complete stroke of luck that a very observant scientist just happened to notice. Now, check out this timeline, because this is where the story gets really interesting. The idea wasn't totally new in 1928. Way back in 1896, a guy named Ernest Duchenne actually saw mold killing bacteria, but his work was completely ignored and forgotten. Just vanished. Then you fast forward 3 decades to 1928. Alexander Fleming comes along, goes on vacation, leaves a messy lab, classic, and comes back to find that a stray bit of mold, Penicillium notatum, is wiping out the bacteria on his petri dish. That's the famous eureka moment. But even then, it wasn't a done deal. It took another team, Florian Chain, until 1943 to finally crack the code on how to make enough of it to actually use.
Okay, so Fleming saw that it worked, but the big question is, how does it work? Let's get into the science of it, because penicillin isn't just a poison. It's way smarter than that. It's like a brilliant little saboteur. So here's the secret weapon, a little chemical structure called the beta-lactam ring. This ring is the absolute key to everything. You can think of it as a master of disguise. It looks almost exactly like a protein that bacteria desperately need for a very important job. And here's its 3-step plan of attack. First, that deceptive beta-lactam ring latches onto the enzyme the bacteria uses to build its cell wall. Step 2, it completely jams up the works, so the bacteria can't build new walls or even repair its old ones. And then, the final step, with its protective wall compromised, the pressure inside the bacteria builds and builds until, pop, the cell literally bursts. And the best part? Our cells don't have cell walls, which is why penicillin is so deadly to bacteria but totally harmless to us. So figuring out how it worked was a huge breakthrough. But producing it in large quantities, that was a whole different ballgame. And there's nothing like the pressure of a world war to kick innovation into high gear. Yup, World War II is what really took penicillin from this interesting lab discovery to an absolute world-changing necessity. Suddenly, you have millions of soldiers getting wounded, and infections are rampant. The demand for this new antibiotic went through the roof, and that forced scientists to figure out fast, how to make tons of it.
And boy, did they figure it out. So let's take a look at how we do it today, because the journey from a single petri dish to these enormous 50,000-liter vats is just incredible. Okay, so let's start at the beginning. Back in the early days with Fleming's original mold strain, the yield was, well, it was tiny. For every liter of broth they brewed, they got just a single milligram of penicillin, 1 milligram. And today we get 50 times grams per liter, not milligrams, grams. That is an absolutely mind-boggling increase. Let me show you what that actually looks like. I mean, just look at that, a 50,000 times increase. That's not a typo, 50,000x. That is just an unbelievable leap. It's truly one of the greatest success stories in all of biotechnology. And that massive jump, that's the reason penicillin went from a scientific curiosity to a drug that's in every hospital in the world, saving lives every single day. So how in the world did we get here? Well, it wasn't one single thing. It was a whole series of breakthroughs. First, we found a much better strain of mold, Penicillium chrysogenum, and then we improved it even more with genetic tricks. We figured out the perfect food for it to grow in, and we invented this super efficient production method where we grow the mold in these huge liquid tanks and carefully feed it nutrients over time to really maximize how much penicillin it pumps out. So the whole industrial process, you can really split it into 2 main stages. You've got upstream processing, which is all about growing the fungus and getting it to make the penicillin. Then you've got downstream processing, which is the tricky part of actually getting the pure drug out of that messy soup.
So for the upstream part, it all starts with a little batch of fungal spores. They're put into these gigantic, sterile fermentation tanks full of a nutrient-rich liquid. For about 6 days, the fungus just grows and grows. And remember that fed batch thing? That means we're slowly dripping in more food and key ingredients the whole time, which kind of tricks the fungus into working overtime to produce as much penicillin as physically possible. All right, once the fungus has done its job, we move downstream. First, you filter out all the fungal cells themselves. Then comes the really clever chemistry. You use a solvent to pull the penicillin out of the broth. By changing the pH level up and down, you can make the penicillin literally jump back and forth between the watery broth and the oily solvent. And each time it does, it leaves more impurities behind. It's brilliant. Finally, you're left with a pure product that you can turn into a stable crystal salt.
So we've seen how it's made, but here's another cool thing. By tweaking that process, we can actually make different kinds of penicillin, because, as it turns out, not all penicillin is the same. The two big ones are penicillin G and penicillin V. And here's the crucial difference. Penicillin G gets destroyed by your stomach acid, so you have to get it as an injection. But penicillin V, it's totally stable in acid, which means you can take it as a pill. And the only real difference is a tiny tweak to one of the precursor ingredients you feed the fungus during that whole fermentation process. Pretty neat, huh? So there's absolutely no doubt about it. Penicillin was a game changer. It kicked off the age of antibiotics and saved literally hundreds of millions of lives. But today, we're facing a new, scary problem: antibiotic resistance, where bacteria are evolving to fight back against our drugs. And that leaves us with a huge question. What's the next penicillin going to be?